With this talk, I would like to do two things. First, a little bit see something that is a little bit complementary, and Peter mentioned it already, with what has been discussed in terms of functional imaging, and go back to structure, and more specifically to microstructure imaging by diffusion MRI. And also, since we are celebrating the 20th anniversary, I would also like to take a few minutes to just draw a little bit of perspective on what we have done over the last 20 years in the context of the CIBM, which has been, you know, the catalyzer for this research. So my lab is indeed at EPFL, mostly, with a joint affiliation with the CHUV, UNIL, and the CIBM. And we work mostly on computational medical imaging, which means the computational tools and the methods that we develop and apply to acquire, reconstruct, and analyze medical images, with a specific emphasis on inverse problems, as you will see, and applications to diffusion MRI, but also to ultrasound, digital pathology, and more. And we have another subgroup in the lab doing more computer vision and machine learning. So what I would like to discuss with you today is really the work that we have done and the perspectives in diffusion MRI for microstructure imaging. And diffusion MRI, well, it's quite impressive to talk here about that in front of Professor Le Bihan. But diffusion MRI exploit the very well-known physical principle of diffusion of water molecules in this case, which we all know. Water molecules diffuse in a medium with a random motion that has been described in statistical terms thanks to the discoveries of Einstein at the beginning of the previous century. And so we model the diffusion in statistical terms. What is the ability of the water molecules to move in a medium? How far can they go? In which direction can they go? We model that with a distribution of probability. And if we study this diffusion of the water molecule in living tissues, which is what we do in our work, this is very interesting because it tells you a lot about the architecture of the tissue. And indeed, if you consider, as a typical example, the cerebral white matter, the structure of the tissue itself restricts the diffusion because the molecules are not able to diffuse freely in all the directions and to all distances because they are constrained by the boundaries of the tissue they live in. And this is exactly what is exploited in diffusion MRI. We try to measure this ability of the water molecules to diffuse in distance and in directions at every point of a medium, typically white matter here, to really learn more about the structure that restricts this diffusion. And indeed, the white matter has been one of the very first targets for this research because it is highly restricting the diffusion of the water molecules. If you take a point in the cerebral white matter, what you will see is that it is made of tightly packed axons surrounded by their myelin, which really restricts the diffusion. And specifically, it restricts, do I have a mouse? Yes, kind of, yeah. It restricts the diffusion more perpendicular to the axons than parallel to them because the waters are reasonably free to diffuse parallel to the axons, but they are restricted by the boundaries very quickly perpendicular to them. And so if you consider the typical medium-like parallel bundles, parallel axons forming a bundle, you will see that there, the distribution of probability of the diffusion shows this structure of the tissue in terms of main direction of the bundle. If you look at a more random medium, we'll have a more isotropic, roundish distribution of probability. If you look at fiber crossing, you will see in the diffusion probability, assuming that you can measure it, you will see the structure of the tissue very, very clearly or quite clearly. And this is really the origin of the connectivity, brain connectivity analysis by diffusion MRI, because thanks to the pioneering works of Professor Le Bihan, we know that we can actually measure and model the diffusion in the brain by diffusion magnetic resonance imaging by making the signal that we measure with the MRI, classical signal that we measure with, for instance, a spin echo sequence, make it sensitive to the diffusion of the water molecules at some point in some direction. So we basically get an image which is a kind of a normal MR spin echo image, but where the contrast is actually affected by the ability of the water molecules to diffuse in some directions. And without going too much into details, we can, for instance, make an image that is sensitive to the ability of the water molecules to diffuse, say, in this direction. It's not a 3D perspective, it's really this direction. And if the water molecules are able to diffuse, there will be a drop in the signal that we measure. If they are not able to diffuse in this direction at a given point, there will be no drop. And we can see, indeed, around the corpus callosum, for instance, that on that side, the signal is dark. It's not because the patient has a stroke, it's just because there are fibers in that direction here that are parallel to the direction that we measure. Where is my mouse? Here. And so since the fibers here are mostly parallel, it's difficult to move the mouse, sorry, but you see it moving a little bit, parallel to the direction that we measure the diffusion of, then we have a drop in signal. While on the other side, where is it? Here, you see that the fibers are like that, if you see my mouse, more or less, perpendicular to the direction that we measure, so the diffusion is restricted, there is no dropping signal. That's the basic idea. And so with that, we can actually probe at every point of a volume whether the molecules are able to diffuse in a large number of directions, and from that, reconstruct at every point of a brain or of an organ the probability of diffusion, and therefore measure something like initial works were really dedicated to measuring the fiber orientations because we measure restriction of diffusion in some directions and ability to diffuse in other directions everywhere. And so initially, the work in brain connectivity analysis by diffusion MRI really led to collecting this kind of information where at each point of, say, the white matter, we measure the probability density function, or a simplified version here that I generally call the peanuts model, of the ability of the water molecules to diffuse, which reflects the orientation of the fibers locally. You can navigate those peanuts within the brain, and from that, what we worked a lot with on is to infer brain connectivity, try to link the dots, and this is really the pioneering work done with my former PhD student, now professor at CHUV, Patrick Hagman, in collaboration with Professor Meuly, really trying to show that we can have a quantitative assessment of the brain connectivity between regions, and this is really concomitant with the appearance of the CIBM. This work is exactly 20 years old. So this paper was published in 2006, the other one was published in 2003, so very initial works on tractography showing that we can really build a map, a quantitative map of the brain connectivity that can be used to assess problems in connectivity. And this analysis of the whole brain connectivity by diffusion MRI has been named connectome. The connectome is the set of fibers. And this very term, connectome, has been actually invented very much at the same time by Patrick Hagman and by Olaf Spons in the US. So the term connectome that is now widely used in the world is actually one of the subproducts of, say, the CIBM peanut. So we can be proud of it. And you can double check. This term has been indeed co-invented by Olaf and Patrick pretty much at the same time, and at that time independently, although we have been working together later on. Well, this idea of connectomics, connectome analysis, then made its way through the clinical investigations, and this is just a very quick and not updated summary of clinical investigations where we try to show indeed that disruption in brain connectivity can be a sign of some of the most prominent brain pathologies, and I just signed a very non-exhaustive list of papers that we have co-authored showing that this kind of technique can be used to investigate pathologies. And I would like to acknowledge again, thanks to the CIBM, the collaborations that we have been able to establish with the CIBM partners, namely, for instance, for brain development with Geneva, the group of Petra Huppi. For schizophrenia, I would like to say hello to Kim Do here. We did a lot of work with Patrick Hagman, Philippe Conus, and Kim Do on schizophrenia and brain connectivity. Epilepsy is obviously in Geneva with Margita Seeg and Serge Villemot. Hello, Serge, you will be a speaker later today, and so on and so on and so on. So this kind of technique, brain connectivity analysis, made its way through clinical research at least, if not clinical diagnosis. I would like to now switch gear and talk about the recent work that we have done. This is history made possible through the CIBM. Now, what are we working on now in the lab is a little bit different. We are a little bit going away from tractography itself to investigate more the nature of the diffusion signal. And the key hypothesis that we have, or the key assumption that we take, is that the signal that we receive tells us much more than the orientation of the fibers. Actually, it tells us a lot about the microarchitecture of the tissue. The idea is that the signal that we receive is actually composed of all the little contributions to the signal by the different components of a real tissue. And if we measure a diffusion signal at some point, it's actually a combination of signals that reflect the fact that they come from very small restricted regions of the tissue, very restricted signal. Some of them come from less restricted, hindered signal between, so you recognize here, of course I should mention it, but you recognize here an histological section of in the white matter. And if we consider a huge voxel, a voxel in MRI is big, it's 1 millimeter square to say the least. What the signal that we receive from this big voxel is actually the sum of little contributions of different types of diffusion patterns. Some of them show restriction of the diffusion, some of them show just loose restriction, hindered diffusion, and some of the points in my volume show pretty free diffusion between the axons. So this is what we actually get. The signal is much more rich than just saying there is restriction. There are different types of restriction. And the goal is to say, from this big voxel, we receive one single big signal, well, several, but say few signals. Can we use that to finally understand the microstructure? Try to determine how much, what volume fraction of the voxel is made of intra-axonal compartment, extra-axonal compartment, free water, big axons, small axons. And so very quickly, a few years ago, we came up with this framework of inverse model. From an average signal, can we reverse it to explain it by identifying the parameters of the tissue at the micro level? And that's really the idea of having computation tools, inverse models. So basically, we have two parts in that work. We first have to understand, and I have to speed up a bit, I know. We have to understand the forward model. If we have a tissue, what is the signal that we would get? The forward model. This is simple or difficult. It can be very simple if the tissue is modeled by a very simple model. I will come back to that. But it can be very complicated if we model the tissue as a very complicated structure. So understanding completely the forward model is really the first step. Because when you understand that, the goal, of course, is then to take your MRI signal and reverse it to identify the parameters of the tissue that best explain your MRI signal. And this is the second part, which is the inversion, and there are computational frameworks as well to do that. And the initial work that we have done on that, to show it, is really to say, okay, let's take a very simple model of the white matter tissue, like parallel axons, parallel tubes. Tubes of different radius, but simple parallel tubes. Not more complicated than that. The only parameter was the mean value of the axons within the voxel. So is my voxel made of big axons or small axons? It's very coarse, but let's take this. And if we do that, we can, by analytical calculation, we can generate examples of signals that would correspond to different types of tissues, different types of axonal diameter. So we can generate a dictionary of signals, each of them being the signal that would be generated by following the physics of MRI, the signal that would be generated for different types of tissues. And then we try to find the one or the ones that best explain my tissue. And this is the work that we did with my PhD student of that time, Mohamed Barakovic. So, but you can kind of see the picture itself already. So we managed to build a map, which is not an fMRI map, actually. It's a map of the mean axonal diameter of the axons within each voxel. So the color coding is actually the mean value of the axonal diameter for that, highlighting that the motor cortex is predominantly made by bigger axons than the other parts of the brain. So you see them in color. They are just here, just the callosal connections, not more. But you see that we can map things that are at the micro level. Axons have a diameter of a few microns, by measuring a signal at the millimeter level, by inversing that so that we find the microstructure parameters that best explain my macro signal. So at the end of the day, we come up with this framework where we have, and it might look a bit complex, but it explains the work that we are doing now. What we want to do is to study patients through MRI. And what we do to do that is to build synthetic model of the tissue we want to understand, simple or more complicated models, derive the signals that we would get for these types of theoretical tissues, so that when we have the real patient, we get the real signals and we are able to identify, recognize which type of tissue would have the highest likelihood to generate this signal. I make it a bit simple, but that's the idea. So basically, from the dictionary of possible signals, we try to identify the ones that correspond to the real signals so that we can identify the microparameters, microstructural parameters of the real tissue. Then we can use them for further studies. We obviously have to validate those assumptions, and we can iterate that. And I would like just to now spend a few minutes, not too many, I promise, on the different steps for completing this global framework, because it involves quite a lot of computational research. The first one is to be able to generate the MRI signal from a known tissue, to have a way to say, if I have a tissue, this is the diffusion MRI signal that we would get. If the model of my tissue is very simple, I can solve that analytically by solving the block equations and showing that for a tissue made of simple tubes, I can generate the exact formula, the closed form expression of the tissue, the signal I would get. But as soon as I come to more complicated tissues, I will not be able to solve that analytically. And so the first piece of this picture is to build a simulator. And we have built, that is the work of Jonathan Patigno. Johnny is here somewhere. He did his PhD mostly on many things, but mostly on building this simulator that is really a very high-end simulator that is used by many groups in the world today, because it can really generate extremely precise simulations of the diffusion of water molecules in a given tissue and the corresponding MR signal if you consider a given MR sequence. So this MCDC simulator has been published, it's available, and it is used by actually many groups around the world to generate the prototypes, the possible tissue signals that would correspond to a tissue. So that work of Johnny is a very important part of the puzzle that allows, for instance, to study many things in terms of what we can expect to read in a signal. For instance, we can say, what if we decrease or increase the resolution of the voxels, the size of the voxels? Can we see more or less? We can simulate that very, very precisely because we can make synthetic substrates, then downsample and simulate exactly what we will get as a signal. So understand completely what we can extract and what we cannot extract with that. We can also say, what if in the tissue there is a loss of myelin, so an increase of permeability? What will change? Are we sensitive to that? So we can really simulate all this to really understand the forward mode, the forward path. And more, what is the effect of the size of the simulation and things like that? I need to speed up, so I will not spend too much time on that, but we have Johnny is here and we can discuss that in detail. Next step is to generate possible configurations of tissue. We have to go beyond the simple tubes, obviously. What we did is to really go in the details of the neuroanatomy. And we work with neuroanatomists, and we actually investigated in detail the actual axonal geometry within, in this case, rats, to really understand what are the parameters that could matter. Diameter, but also ondulations, change in diameter, dispersion of a bundle, things like that. And we did that through synchrotron imaging. So we did experiments with the synchrotron facility in Grenoble in collaboration with our colleagues at DTU in Copenhagen. And we published that paper showing the statistics of the different geometrical parameters of the axons. That would allow us to build synthetic phantoms, because the next step when we have done that was published, as you can see, 4 years ago already, then came my next PhD student, so Juan Luis Villarreal, who built Cactus. He's Mexican, so he had to build cactus. And he built cactus, which is a synthetic white matter generator. So he built a software framework to really build very sophisticated synthetic phantoms of architecture that you can vary by setting the parameters of density, distribution of accents, undulation, dispersion, whatever. Density, I said it. And he managed to build that. And we have today, Cactus is today the system that is probably the most advanced in terms of generating realistic configurations for white matter, because it allows to generate, for instance, very high accent densities. The nature of white matter in some bundles is that the accents are very packed, occupying something like maybe 90% of the volume in some places, and none of the existing configurator were able to reach that packing density, and also to simulate voxels as big as alpha millimeter, which for numerical simulations, remember that the resolution is the resolution of an axon, so we have to model individual axons and group them in alpha millimeter, which is huge, 3 orders of magnitude bigger than the axons themselves. And so we can generate axonal phantoms as big as that one. We can validate that indeed we can generate them and they correspond to the correct distribution that we measured in the synchrotron imaging. And then we can compare with histology. This is a work that we have done in collaboration with the colleagues in Canada of Nicolas Tikoff, showing that indeed if you ask, say, a volume fraction of something, you get it and you can compare to an histological section where this volume fraction is also present. If you ask a myelin density of so much, the generator will give you, and you can compare with real tissue, and you have a pretty good match of what we want with respect to what we measure in histology. So the simulator is validated. It allows to generate many examples. It takes time because it's not computationally light, right? But you can generate variations of radius, variations of dispersion, variations of volume fractions, and generate those prototypes and then simulate them to build your dictionary of very sophisticated configurations of white matter. And you can, by the way, as a side product, generate very realistic phantoms to validate tractography. So if you go back to tractography, you can generate with Cactus very, very fine grain phantoms, and we did it. This is the DISCO data set that is now widely recognized. But you can also use that to close the loop and have your atoms, your prototypes here that will help you identify the real one. And I just want to finish by mentioning a few more contributions that are now early stage in the lab. When we have done that, we can also work in optimizing the MR sequence. What is the MR sequence that will, beyond the classical one that we use, the PGSC that we use, what are the sequence that would be more specific or more sensitive to some parameters of the white matter? And this is the work that is taken care of by my new PhD student, Ekin. She's also here somewhere, in collaboration with the colleagues in Rennes, in RIA. And then comes the application of that. We have different paths of application in the white matter, of course, but I just would like to mention the work of Elda Fischi. She's also here. Where are you, Elda? Over there. So Dr. Fischi is a postdoc in my group and a CIBM staff member, and she has been studying brain development for years, and you have seen the flagship project is tightly related to the work of Elda. And she has been studying the development of the brain connectivity for many, many years. You see one of the first papers there 10 years back. But now she just got a new project funded by the National Science Foundation, and the PhD student working on that is Andres, to study now, with the microstructure imaging that we have developed, the development of the cortical gray matter. So the cortical columns and the development through time, through gestation and childhood, to identify normal and abnormal development paths at the level of the cortical columns. I will not go in more details. If you want to know more, Elda is over there. What else? What else? Well, why only staying in the brain? Why not going outside the brain? And we now have a very important track in our group to work in oncology, not in neurology anymore, but in oncology, to study cancer. And the idea is very much the same. Can we have synthetic models of tissues where we identify what we want to measure to be sensitive to the early appearance of cancer, typically invasion of cancerous cells in an organ? Can we model that? Can we simulate that? Can we identify that from the real signals and give a very quantitative assessment of the distributions of cells in a tissue, very much like what you would do with a biopsy, but without taking the tissue, and having a much broader picture of what we can do. So replacing the very invasive biopsy by a virtual biopsy. And we do that in the first case in lymph nodes. Lymph nodes are very important targets because they are the primary target for many cancers. And we do the same. The problem, the difference with the brain and the white matter specifically, is that in other tissues, you don't have the myelin. So the water molecule diffusion, the diffusion of the water molecules is much less restricted. And much less restriction means more problems for us. And so we have to change the model. And my PhD student, Rémi Gardier, I say doctor between parentheses because he had his private PhD defense, but not the public yet. So he's not doctor yet, but he's half doctor. So half Dr. Gardier built a model for lymph node diffusion MRI. Very much you see the simulation for the white matter. You see the simulation on the bottom here for typical lymph node tissues, cancer and non-cancer. Very much the same story, except that we now have to take into account the fact that the tissue is very permeable. And to make a long story short, Rémi built a full model of the diffusion signal accounting for permeability, which is able for us, when we have a real signal, to really identify those parameters like the radius of the cells, the permeability, the diffusion coefficients inside and outside the tissue, so those parameters that you see them. And we did that. We did that in simulations, showing that indeed permeability identification matters. And we did that in real tissue, and this is my two last slides. We are now running real experiments with the help of many people in the CIBM constellation. This is the work that we are doing with Professor Ilescu. Liana is somewhere there. Thank you very much for your help and very fruitful collaboration. On lymph nodes of rats, where we managed to get them, to scan them in the CIBM scanners at very high field, and to identify those parameters that are in the model, like the radius of the cells, the permeability, the volume fraction, and so on. This is very preliminary unpublished results. We still very much have to go in more details. This is just one example. And this is done with a medical student, Arnaud, also. And then, not later than last week, 2 weeks ago probably, we started with human experiments. And I will not be long, but it is a very long process, as you can imagine, because we have to control the ethical aspects, of course, obviously. And moving to human patients is not an easy step. It took a while to get the ethical clearance. But two weeks ago, we managed to have the first lymph nodes of healthy patients. It might look strange to remove lymph nodes from healthy patients, but actually they were patients operated for something else than a potential cancer, for which some of their lymph nodes in the abdominal cavity were anyway removed, and so we managed to get access to them, to transport them from CHUV to EPFL, to scan them, to bring them back to pathology, and to have the confirmation that they have not been altered by this process, so that ultimately they could still be used to diagnose cancer, so we are on the safe side in terms of ethics. And I have no more picture than that, just show you that we got them, which is, it doesn't look impressive, but it's quite an achievement, I can tell you. And so this is the work of Rémi with the medical student Arnaud and my new PhD student Salomé Bob. She's also around and be able to discuss. And that's the end of my talk. I'm sorry I've been a bit long also. This is the result of a very long journey, started very parallel to the CIBM and in the context of the CIBM. So I want to thank my team. I want to dedicate this speech to Professor Innocenti, who passed away a few years ago and who was, at the end of his career as neuroanatomist, part of my lab, and I'm extremely proud of that. And my former and current postdocs, you recognize the names there of those that you have seen on the slides. My former and current PhD students, former members of the lab, the very fruitful initial collaboration with Patrick Hagman and Reto Meuli, and the ongoing collaboration with Ileana, of course, but also with the clinical teams at the CHUV, Clarisse Droma in radiology, Christine Sampou in pathology, and the team in surgery, who really operate the people, so Professor Hanloser and Dr. Chapalet, who really operate and give us access to this incredible material. And last but not least, the CIBM team under the leadership of Pina, with the extremely helpful collaboration with Bernard and Kasia. Thank you to both of them also for that. Thank you very much.